All right, so thanks everyone for joining us today. I hope you all had a great week. So today we have the pleasure to host Jack, who is a PhD student with Danilo, and he's gonna share with us some thoughts on attempts at LLM explainability in biomedicine. So before handing the mic over to you, Jack, let me remind you all of some house rules. As usual, clarification questions are allowed to be asked during the talk. For people in person, just raise your hand and wait for me to give you the mic so that people online can hear you. And for people online, you just have to type the word question. I have a question in the chat, and when the time is right, I'll let you directly ask it. There'll also be some additional time at the end of the talk for people to ask further questions, make some comments, and have a more informal discussion with Jack. That's pretty much it for me. So whenever you're ready, Jack, floor is yours.
Perfect. Yeah, thanks a lot, Rim. So thanks everyone for coming in person or joining online. So as Rim was saying, I'm going to outline some of our recent practical attempts at language model explainability in biomedicine. And in particular, we're going to look at a specific use case of health records in autism. So really, I want to divide this talk into 3 sections. So the first one is just really more of a general overview of some ways of thinking about transformer interpretability that sort of motivated some of our early analyses. And then I'm going to outline a bit more in detail the specific case study that's centered on autism diagnosis. And then finally, get into more detailing in much more detail the modeling techniques that we use to adapt these transformers for more clinically relevant insight. So just a quick plug for our lab. So we are a highly technical team at the intersection of neuroscience and machine learning. And we really try to have equal footing in the research objective and the research method. And what that usually works out to is developing and implementing very custom tailored approaches using machine learning and deep learning tools to get at the biomedical research questions at hand. So getting to the meat of the talk, what do we exactly want from our AI models in the clinical setting? Well, there's really 3 things that we want here. The first is, of course, excellent performance on the task at hand. Does our model actually learn the task relevant information? But almost equally important is, we want some sort of human interpretable task explanations from the model. So, can we actually identify, what it exactly is that our model learned about the task? And the third point, might not be applicable in all cases, but it's certainly very relevant in a clinical setting, is the ability to interface with multiple sort of different information sources. And what I mean by this is that, is it possible to use our model to maybe probe existing clinical standards and compare them to what our model found, which may enable us to suggest revision to these existing clinical standards. So in terms of transformers, we've all seen empirically that the first criteria is certainly met. We've seen amazing prediction and inference capabilities with transformers, but the second 2 criteria are a little bit more challenging, of course, and that's really the subject of today's talk. So why did we want to use LLMs for clinical insight in the first place? Well, the main thing is that unstructured text is actually quite abundant in our healthcare systems in general. We might have a lot of people have this idea that healthcare is very structured and regimented and there's lots of quantitative metrics, but a lot of the communication and the diagnostic processes, and this is particularly the case in psychiatry, where these unstructured clinical documents really form the foundation of the diagnosis itself. But fortunately, for the first time, as we've seen in the past couple of years, we have amazing language models now that can generate, re-express, and evaluate human language at near human levels of capability. So really the secret sauce that makes this directly applicable to biomedicine is transfer learning. So the paradigm here is that with pre-trained models, the advanced language skills that we need to carry out the task already come built in. What this allows us to do then is that we can fine-tune on these pre-trained models to learn much more fine-grained and task-specific information that we actually care about. And with this paradigm, we've effectively separated the task of modality learning from the task-specific learning that we actually want to investigate. And fortunately, transformers have exhibited unprecedented transfer learning capabilities. And this is especially important in biomedicine because in most cases we just don't have large data troves of like this internet scale data in other disciplines of machine learning that to train our models on. So we've seen empirically that fine-tuning is effective with as few as 1,000 label training examples. And this enables us to ask questions that would have been impossible to answer previously if we had to train models from scratch. So this is a very, I think, illustrative example. This is using a pre-trained model, just pre-trained on human language for a Python language prediction task. And you can see that in the fine-tuning regime, which is this blue curve here, you need orders of magnitude less task-specific data than just training from scratch. And this is, of course, will carry over into the biomedical domain as well. So now looking at the internals of these models more deeply, how can we actually deconstruct the basic building blocks of these transformers to reveal more mechanistic insight into the internal operations that lead to inference and prediction? So this is an example of the encoder block of the transformer, and I'm just going to focus on the encoder here because that's the model that we use in our analyses. But these building blocks are, of course, applicable in the decoder and the encoder-decoder regime as well. So really, I'm going to focus on 3 specific areas in this overview. The first is the input embeddings of the actual input itself, then moving on to the attention block, and then finally the feed-forward network or multi-layer perceptron. So looking at the embeddings, first off, so these are perhaps the most important development in language modeling in the last couple of decades. It's just that we can represent words and other semantic elements like sentences or documents as real valued vectors. And with these embeddings, we can really capture semantic information in this vector format via training. This is a very simplified visualization of word2vec, just inputting semantic elements like words and outputting some vectors with some relational meaning to each other based on some pre-training objective like mass language modeling or some other training method. So for biomedicine specifically, the prediction itself isn't usually the main goal. Instead, this prediction problem is usually used as a proxy for learning the proper representations of the embeddings that are most useful for other tasks, right? And you can see here, these are just examples of some embeddings and some stylized examples of some embeddings in vector space. And the idea here is that these embeddings are very, question?
Thank you. Yeah, could you just talk a bit more about what kind of task you will be considering here? What would be the setting, just to get a better sense of what is it we would like to get at the interpretability?
Yeah, sure, for sure. So, a very simple example is just like diagnosis classification. Do they have the disorder or the disease or not? And that would be, the training objective there. So in the biomedical case, these embeddings would take on some sort of semantic information relevant to the diagnosis, for example. So that's sort of the setting that we're talking. And I'll go more in detail in our case study.
Perfect, thank you.
Yeah. And typically these embeddings are readily interpretable. You can do various vector operations with them that have semantic meaning. You can look at the similarity between different embeddings using things like cosine similarity. That usually does have, empirically, we found has a very good meaning associated with it that's easily interpretable. And moving on a little bit, so this sort of motivated some of our early work. This is a sort of a formulation of the transformer architecture that was presented by the Anthropic interpretability team. And this is what they termed the residual stream. So essentially, we have input embeddings that travel through the architecture. They travel along this residual stream, and then they're diverted right into these attention modules or MLP layers, then transformed by these layers, and then added back in at certain points. So really, this is just like feeding in embeddings, performing some transformations on them, and then putting them back into the residual stream. So what this perspective allows us to realize is that the dimensionality of these various attention components is actually usually significantly smaller than the embedding dimensionality itself. So this is very common in basically all transformer architectures now. And what this means is that each individual attention head would operate on comparatively small subspaces of the residual stream itself. So this gave us the original idea to maybe factorize these in some way. And the important part here is that even these subspaces have very low rank structure. So we could maybe factorize these and identify the most important components here for interpretability. Now, also, again, the function of this attention mechanism is usually to mix different information between the residual streams of different tokens, right, which is visualized in this bottom picture here. And one of the difficulties, though, with this framework is that layers in the network can actually send different information to subsequent layers by storing it in different subspaces to be accessed later. So this makes it actually a bit more challenging for interpretability because you sort of have to consider all the layers at once instead of just looking at a single isolated layer. Now, another way to think about this attention module is decomposing it into two separable linear operations or circuits. So the first circuit here is the query key circuit, which essentially just controls which information a specific source token would attend to. And then the output of value circuit determines how you would transform that information that you're reading and how to incorporate it back into the residual stream again. So this is a very nice formulation because essentially we've reduced this complicated attention mechanism down to two separable linear operations. But still, interpretation with this is not exactly trivial because what we have now is really essentially just a large generalized linear model that acts upon many highly correlated input features, which are the embeddings, of course. And that's still not super easy for interpretation in the best case scenario. Now, getting to the multilayer perceptrons or the MLPs now. So these are probably the largest stumbling block to interpretability at the current moment. And these are not, we can't just ignore these because although the attention mechanism is essential to the functioning of the transformer, about 2/3 of the transformer's total parameters are actually contained within the MLP. So they're obviously very important for inference, of course. And these MLPs are inherently very difficult to interpret due to polysemanticity, where individual neurons can respond to multiple unrelated features. And the reason for this is likely a byproduct of sparse coding, which is where each layer is capable of representing more abstract features than neurons. So, and by features, I mean not necessarily like the input features, but just like more abstract features, like in case of vision models would be things like edges or curves or things like that. In transformers and language, it might be more abstract notions like, I don't know, maybe directionality or something like that. But ultimately, this phenomenon is known as superposition, where intermediate features are spread across multiple neurons. So the trouble here is that you can't attribute one specific feature to one specific neuron. You have to sort of take them all at once. And another way of formulating sparse coding that is a bit easier for interpretation is to treat each neuron as a feature. And then instead of treating each neuron as a feature, we can define an activation space, which is all possible linear combinations of these neuron activations. So in this space, these individual neurons, instead of being features, correspond to basis vectors in this space. And then if these representations align with these basis directions, then they could be considered to be corresponding to a privileged basis, which is much easier for interpretability because if your feature aligns with the basis, that's simply much simpler to interpret as linear combination of feature activation or of neuron activations. So the issue here, though, is that most feature representations don't align with the privileged basis. As you can see in the superposition cartoon here on this slide, if you have two basis vectors, which are the orange dashed lines, and three of these feature vectors, there's really no way to align these with the basis while preserving the relationship between them. But perhaps we could, and this is something that we didn't attempt, but we would very much like to look into further, is that perhaps we could decompose these major factors of variation within the feature representations to better align those with the interpretable privilege basis. So instead of considering just three features, we could maybe identify like two components of these features that are most relevant. So shifting gears a little bit to the actual case study, I'm just going to motivate a little bit why studying autism is interesting from biomedical and from an interpretation perspective. So For psychiatric diagnosis more generally, unlike most areas of medicine, or many areas of medicine, I should say, diagnosing these psychiatric and neurodevelopmental disorders like autism is not exactly a hard science. Like, you can't take blood samples, you can't look at specific biomarkers in order to diagnose. Instead, you have to rely upon these more qualitative descriptions and the interpretation of patterns of behavior of individuals in order to classify patients into certain disorder categories, right? And ultimately, the final diagnosis rests on clinical judgment, which is a very important notion that I'm going to return to very shortly. Now, just a brief overview on the history of autism, of the term autism. This was originally coined in the early 1900s by Bleuler, who was a contemporary of Sigmund Freud. He did a lot of work on schizophrenia, and he used this term to describe a particular symptom of some of the most severe cases of schizophrenia, in fact. But of course, by the 60s and 70s, this term had morphed to focus primarily more on social deficits, which is much more aligned with our modern conception of the disorder. But since then, still, these diagnostic instruments that clinicians have used have undergone significant revision since then to include things. And there's been quite a lot of debate in the autism research community about what things should actually be specific to autism and are most diagnosis relevant. And this is all to say that this issue of specificity has really challenged autism since the very beginning, or the very inception of the term. So the idea here in this study is really, how can we place notions of autism on a much firmer grounding? And currently, autism is very interesting to study because it has a prevalence of around 1 to 2% in the general population, which is significantly higher than other neurodevelopmental or psychiatric disorders. And interestingly, while autism is known to be highly genetically heritable, there's been very little progress in identifying like autism-specific genetic pathways or diagnostic indicators or biomarkers. And brain imaging studies have also not provided very much insight into the actual biological factors that drive autism diagnosis. So currently, again, this is the only effective means of diagnosing autism is still clinical judgment, which I'll just say has been remarkably consistent and reliable across clinicians for several decades. So how does this diagnostic process actually work? So first, children are usually observed and interviewed, if possible, because they may be nonverbal or might be too young in order to have speech. And they're usually done alongside parents, by an experienced physician here. So most physicians follow the DSM-5 manual, and the DSM-5 contains various checklists of behavioral criteria that are supposedly most indicative of autism. Now, of course, there are also additional diagnostic instruments that some physicians use, but ultimately the final burden of diagnosis falls on the clinician's own intuition and judgment. And this is just based on their assessment of their actual observations and the analysis of their provided developmental history. So the idea here is, what if we could actually deconvolve this clinical intuition to better understand autism and autism-specific behaviors? And to do this, we would analyze written clinician observations, and we could maybe peer into the mind of the clinician by doing so. Simply because these written notes actually contain, by definition, the highest intrinsic density of diagnosis-relevant information, because this is what the physicians are actually using to make their diagnosis. And in doing so, we could maybe revisit the idea of these truly autism-specific behaviors, which have been directly identified again by experienced clinicians and written down in these clinical notes. So this was work done with our collaborators at University of Montreal. So these are two clinicians and very experienced autism researchers. They helped us with the collection of the data and also some of the interpretation and guiding us on the analysis approaches that appear to be working to them. So the idea here was to first collect clinical observation reports from the physicians, then fine-tune a pre-trained language model to classify each report based on the final diagnosis. And then we could identify from this language model specific semantic elements that drive the autism classification. And then finally, we could maybe revisit the established diagnostic criteria using these empirical findings from our language model. So getting back to this notion of specificity, this is an important point to stress about these reports, is that these reports come from children with suspected autism already, who have been referred to a specialized clinic in North Montreal. And here, an autism expert, which is typically a psychiatrist with multiple decades of experience, would observe and interview each child and record their observations in a written report, as I said. And these reports can be multiple pages long, so they're not simple at all. And importantly, they contain no diagnosis-specific information. They're all just observational and unbiased reports. And then after this, a panel of experts would agree to a consensus autism or non-autism diagnosis, just based on the description of the child in the report, so using no other external information except these reports. And this is important to stress here, because all these children have been suspected to have autism when they arrive at the clinic, the observations that are contained within each report will contain only the most autism-specific diagnostic information. So this is not comparing children with diagnosed autism to children who are neurotypical. It's comparing children with autism to children who have been suspected but eventually ruled out by these diagnostic process to have autism. So this is a brief overview of the data collection protocol. So first, again, the physician would engage in various activities with the child, and then next, write a report in an unstructured written format. And importantly, these notes don't contain any direct diagnostic assumptions from the clinicians. They're just simply unbiased observational documents that other were scanned and manually anonymized by our collaborators to remove the specific identifiers of the children or parents. Then we used a Google Document AI OCR tool, which was super powerful, by the way. If anyone is working with scanned PDFs, I would highly recommend this tool. It's very, very effective. So this was used, again, to transcribe the scanned PDFs into text documents that our LLM could read. And then we just performed some very minimal text cleaning to remove any obvious artifacts and OCR errors there. So now moving on to sort of the interpretability stage of the study. So this is just a high-level overview of the LLM pipeline. I'm going to go much more into detail in a couple slides of this interpretability pipeline, but just suffice to say, for now, we first started with a pre-trained Roberta-based language model. Again, pre-trained just on general text information. And this formed really the central basis of our approach. And then we added some interpretability pipelines as well, which I'll detail in a bit. But first, the fine-tuning results were significantly superior to what was typically found with biodata. So we got nearly 80% diagnostic accuracy here, fine-tuned on only a little over 4,000 reports. But this might not seem like a lot in machine learning context, but in a biomedical context like this is actually quite a large study. So this is significantly better than, for example, results from brain imaging, which get around 60 to 70% accuracy. And these brain imaging results are typically on neurotypicals versus children with autism, and we're working with a much more difficult to diagnose cohort here. So these are quite good results and show that our model is actually learning some interesting diagnostic relevant information here. So next, moving on to the interpretability, we wanted to try and unpack this decision-making of the fine-tuned LLM. Why did it actually predict one report to be indicative of autism and another not to be? So, of course, a tempting place to start is to look at these attention weight matrices produced by the attention heads in each layer. So this is sort of calling back to what I was talking about in the introduction. Maybe we can decompose these attention matrices, because they have significant low rank structure, into major factors of variation that can give us more insight into the diagnosis. So we tried some unsupervised methods like PCA and non-negative matrix factorization, and also some supervised methods like partial least squares, which is, again, using the supervision signal of the diagnosis itself to present, sort of decompose in a more structured way. And we did this to try and identify essentially which words or groups of words receive the most attention in the model and which, again, are most diagnosis relevant. So at first, this approach produced some somewhat promising, but ultimately, when we showed this to our collaborators, they were somewhat non-actionable results. So I'll show you what I mean on the next slide. So this is just one example of a plot we get from non-negative matrix factorization, and you can see here this component, which is highly weighted in the autism-specific reports, showed some terminology that is somewhat associated with autism, but ultimately, these sorts of single words are not exactly super actionable, right? And they're not super differentiated between those with autism and those without autism. So, moving on to something a bit more advanced here, we tried to look at to identify really chains of words. Oh yeah, sorry, go ahead.
So the clarification question on how you have autism and non-autism in the last slide. There.
Yeah.
This is what would be samples were gathered on the basis of suspected autism. Like, this is contingent on a diagnosis. Yeah.
That's correct. Yeah. Oh yeah, sorry. The question was just for this slide. What do these diagnosis indicators mean? So this is again just the clinician diagnosis that are provided with each report. So essentially stratified by autism and non-autism. So yeah, to better quantify chains of words, right? Multiple words at a time that are most frequently attended to each other. We then sort of frame this attention matrix as sort of a weighted adjacency matrix to construct these network graphs for multiple heads and across multiple reports. And again, we could try and maybe use some network theoretic measures like path length and centrality to identify the most important words and chains of words that are most diagnosis relevant. And again, this approach produced somewhat disappointing results at the end of the day. So as you can see here, if we're looking at, so these are some very high centrality words that we identified, and looking at their nearest neighbors in the network graph, we can see that it actually does make sense. We have things like language associated with development and delay, and behavior associated with things like repetitive and interest, stereotype. So these are very, again, autism relevant. But again, as the nearest neighbors moved farther from the first nearest neighbor, it became a bit difficult to actually understand what was happening. And it was hard to stratify by autism and non-autism here again. So why didn't these previous two approaches work as well as we'd hoped? Well, so the first mechanistic reason, there are mechanistic reasons, obviously. We have multiple attention heads per layer, followed by very polysemantic, as we were saying, nonlinear feed-forward networks. And this means it's very difficult to attribute the responsibility to specific inputs or parts of the network, even if we're given these very tantalizing attention weights for each word, right? And then some broader epistemological reasons are just that we were looking for so it's a very early field of research. Interpretability doesn't really have established paradigms within which these exact quantitative measures make sense. Even though we get very high weights on maybe some NMF components, that doesn't necessarily mean these are going to be actionable and useful for interpretation by our clinicians. So instead, what we should have been looking for is something known as the signal of structure. So the signal of structure is really just the idea that we should be treating qualitative results as first-class citizens. So this is the idea that we don't exactly know, what we're seeing, but we do know that there's too much structure in this signal that we are seeing to be, just random noise. So a great example of this is from Robert Hooke, investigations into cells. So, at this time, people didn't really have a conception of what a cell was, but looking at these things under a microscope, you could see that there's obvious structure here that was not just an artifact of the measuring instrument. But importantly, in order to not fool yourself, you have to know approximately the mechanism by which those structures are generated, right? So in terms of a microscope, things, it makes things that are small appear bigger. So maybe these are things that we cannot see with the naked eye, but these are structures that exist at a much smaller level. So, based on this, we wanted to maybe change our modeling approach to better capture and validate this quantitative structure. So first identify the qualitative structure, then validate it using quantitative methods afterwards. So the key idea here was to change the unit of interpretation that we're acting on. So the semantic elements that these pre-trained language models, the attention modules act on are at the word level, right? But realistically, the smallest semantic elements that clinical practitioners would use to convey these abstract ideas are not actually individual words, but instead individual sentences, right? So we therefore needed a method to identify only the most important sentences for the autism diagnosis. And this, again, would allow us to identify some much more human interpretable structure and then quantitatively validate this afterwards, after we've identified it. So this is a very sort of a high-level overview of our approach. of our modeling approach. So essentially, we'd start at the word level, and this is handled by our pre-trained and, of course, fine-tuned LLM. And then these are mean pooled into sentence embeddings, which are then, again, handled by a single-head attention module that I'll introduce on the next slide. And then based on this single-head attention module, we produced a weighted mean pooled report embedding. And this finally could be handled by a very simple linear classification layer in order to predict the diagnosis. So essentially, to go up each level of semantic complexity, we just mean pool the previous layers embeddings. And these are, of course, trained end-to-end to instill within them some diagnosis-relevant information, of course. So again, very simply, for each report, we split reports at the sentence level and fed the sentences into the LLM. So the pre-trained LLM essentially handled the in-context learning of single sentences at this point. Then the output of that is mean pooled sentence embeddings that we have for each document. And then we applied a single head attention layer, which is much more interpretable than this long multi-layer, multi-head transformer layer to sort of identify the most important sentences in each report. And then based on the importance weights of the single head attention module, we sort of did an attention-weighted average to create report embeddings, which were then classified with our simple linear classifier to fine-tune on the diagnosis classification task. Right? So this allows us really to capture much more meaningful and diagnosis-relevant signal that is ultimately human interpretable at the level of these sentence embeddings. So now, as a sanity check, once we've fine-tuned this model, we can visualize these sentence embeddings in somewhat of an autism-aware embedding space, because again, we fine-tuned on the diagnosis objective. So this embedding space should be instilled with some relevant information about the diagnosis itself based on the embeddings within it. So here we have just a plot of a PCA decomposition of the sentence embeddings. And we can see here each dot represents a single sentence embedding in a much more compact 2D space. And you can start to see some structure appearing where the most autism or the sentence embeddings that are diagnosed autism sort of cluster in one area of this embedding compared to the non-autism sentences. So this is just a very quick visual check that our LLM is learning something interesting at the sentence level. Then we wanted to really sort of trace the information flow in the model. At what layers does this diagnosis relevant information actually emerge? So to do this, we did a global surrogate modeling approach, where you can see with a more detailed model, there's lots of various steps that are somewhat hard to interpret. But with a surrogate model, something like logistic regression, we were just basically attempting to recreate the outputs of our much more complex model using a much simpler model. And we can see something that is somewhat expected here. At the early layers, the diagnosis relevant information is much less. So what we did here was sort of mean pooled again the sentence embeddings from each layer into a report embedding and then classified that at each distinct layer. And you can see at the early layers there isn't as much diagnostic signal that's being detected by the model, but as the information flows through the model and is progressively transformed, it takes on much more diagnosis relevant semantic character at the end. So now looking at the single head attention matrices themselves, so you can see here that these are the most attended to sentences in each report outlined in this black box here, and we can see that these highly attended sentences are actually incredibly autism relevant. And actually, when we showed these to our collaborators, they were astonished at how well this LLM could actually pinpoint specific sentences within these documents that are to them were immediately indicative of autism. So like for example, this one we have things like echolalia, which if you're not familiar with this term is essentially just unstructured vocalizations that don't necessarily have a meaning. And eye contact initiated but not sustained, and then special interest in things like cars, alignment of objects, sorting by size and color. So these are very autism indicative sentences. And you can see that there's quite a wide variety that our language model identifies as being indicative of autism. So we have, again, things like marked interest in trains. That's very different from things like food selectivity, hypersensitivity to noise. But regardless, our model has learned some of this structure that makes things that makes a particular sentence in a report, most autism specific. So looking at more of a macro level at these highly attended sentences, we can see that the most used words by clinicians to describe children with autism are focused really on stereotyped repetitive behaviors and special interests and perception-based behaviors. So we can see things like letters. They have a marked interest in things like letters in the alphabet, flapping, and these mannerisms, echolalia again, and vocalizations. So again, our collaborators thought these were very, very indicative words to describe children with autism. So now what we wanted to do after we had these qualitative results that seemed very promising to us, we wanted to make sure that these were actually grounded in our model's diagnosis prediction specifically. So, which of course, as we saw in the fine-tuning slide, these achieved very high prediction accuracy of nearly 80%. So this is basically asking the question: how do we ensure that the semantic elements that we actually identified using this approach are actually clinically meaningful? So one way to do this is to compare our results to established clinical information that is external to our model. And a perfect source for this external clinical information is the DSM-5 autism criteria, of course. So just a brief overview of the DSM-5. This contains 2 sections of criteria. The first section contains 3 criteria that are relating to deficits in social communication and social interaction. The second, section B, contains 4 criteria relating to restricted repetitive patterns of behavior, interests, or activities. And importantly, in order to, and the DSM-5 recommends that 3 out of the 3 criteria need to be present in to conclude that there's a diagnosis of autism. So these are recommendations. Now, each criterion here essentially contains, 1 or maybe 2 sentences maximum describing a supposedly autism-specific behavior or set of behaviors. So we thought, we could just run these through our model, generate embeddings for these criteria, and compare them to the embeddings that we already have from our reports. So you can see here, this is again just the same plot I showed before of the sentence embedding space, where each dot represents a sentence embedding. And again, now we have the criteria embeddings overlaid on this embedding space. You can see here already some interesting structures start to form where this B1, B3, and B4 criteria are much more heavily on the autism side of the embedding space, whereas these 4 criteria are not. And they're all, again, clustered very similarly together, which maybe suggests they might be a bit semantically redundant, as we'll see. So again, this has 2 benefits, because we can compare our attention-identified important semantic elements with the DSM-5. And on the other side of the coin, we can also evaluate use the cosine similarity to compare semantic content of the attention-identified important sentences from our reports and the DSM-5 criteria. So the cosine similarity really just measures the directionality of the vectors in the embedding space. So similar vectors or similar semantic meanings would have basically vectors pointing in the same direction. Vectors with no meaning would have them be orthogonal to each other, and then opposite meanings would have them be pointing in opposite directions. So we can see here the distribution of the cosine similarities that have been stratified by autism or non-autism diagnosis. There's a very clear difference, as we might have suspected from the embedding plot, between the B1, B3, and B4 criteria here, and the A1 to A3/B2 criteria. So you can see that the B1, B3, and B4 are very, very similar to the top attended sentences in our autism cohort, whereas the other criteria, they're actually not too similar to many things, but if anything, they're on the side of being non-indicative of autism, of children without autism. So these are very, very interesting results here. But we wanted to evaluate how relevant these similarities are actually diagnostically as our final verification step. And to investigate this, we use the cosine similarities as inputs into a predictive model here. So in this case, this was linear discriminant analysis to capture some of the low rank structure again, to once again predict the diagnosis. So now, in this case, each report will be characterized entirely by these 7 cosine similarities, which are between the top attendance sentence in the report and all 7 of the DSM-5 criteria. So instead of representing each report by this long vector embedding, we just have these 7 cosine similarities to represent each report. And the idea here is to ask if there's some diagnostic signal contained within these cosine similarities. And we do find, in fact, that once again, we can predict out of sample the final diagnosis of each report using only these cosine similarities to the 7 external DSM-5 criteria. So you can see here this pink line shows using the most attendance sentence in our report. We get pretty good prediction accuracy using only these these 7 DSM-5 criteria similarities. Whereas if we use some random sentence from a report that is not necessarily autism relevant, we get much worse performance here. And we can see here using looking at the correlations of the LDA scores with these similarities, we can see that B1, B3, and B4 are in the direction of predicting autism, whereas the other ones are actually, if anything, they're corresponding to the opposite diagnosis. So just a brief summary of this study, we found that in contrast to around previous 50 years of autism research, really, we find that socio-communicative deficits in children are not, in fact, necessarily autism specific. Instead, the most autism specific traits in our cohort were stereotyped and repetitive behaviors, special interests, and perception-based behaviors. And really the key takeaway here is that these clinically actionable findings could motivate some revisions to the established gold standard clinical tools in common use in the clinical settings every day. So the idea is not to say that these social deficit indicators are useless. It's just to say that perhaps we're putting too much weight on these compared to the B items. And just as an aside, it's much easier diagnostically to identify the presence of a behavior versus the absence, which would be the case of social deficits. So it's much more diagnostically feasible to assess, essentially what the additional capabilities of autistic children versus essentially what they lack. So that's from a diagnostic point of view. Now, next steps for our mechanistic interpretability, we'd like to really dive a bit deeper into the actual transformer architecture. So we made some attempts at this, but ultimately we'd like to sort of deconvolve these NLP activations a little bit more to address the polysemanticity of these neurons by extracting, essentially, as we were saying in the introduction, the intrinsic latent dimensions from these neuron activations. Again, using things like non-negative matrix factorization or PLS or CCA, and then also using some gradient descent methods to maybe derive some prototypes in the input space. Like what is the most prototypical autism-specific sentence that clinicians use to identify an individual with autism. So we'd also like to look at the unique contributions of attention heads themselves more specifically, doing things like head ablation, contextualization with various input features, and looking more at the internal model representations as well. Then we'd also like to look at some more emergence phenomena. So we did a bit of this with looking at what layer the diagnosis emerges, but we'd also like to quantify, much more fine-grained aspects as a function of things like training epochs or number of attention heads per layer, and easy to identify. And just this final slide here on some takeaways for interpretability more generally. The idea that maybe chasing after maximum predictive performance metrics and metrics early on in the interpretation process, might make it much more difficult to actually maximize the human interpretability. And designing these interpretability pipelines to yield a qualitative guideposts, right? This signal structure idea. Identifying the domain relevant units of interpretation is very essential to the interpretability task. Of course, you can often be led astray if the level of abstraction is just too granular in the case of words, or maybe too coarse if you're just looking at the entire report, for example. And again, we think that using established external information to really validate these qualitative results is a very powerful sanity check here, and is basically essential in the biomedical and clinical domains. So that's all I have for you today. So thanks very much for listening.
Thank you. We have a bit of time for questions. I have a question.
Yeah.
Do you have in the courts cases where the diagnosis is completely uncertain, like two clinicians don't agree?
That's a good question.
On the findings?
So actually, those were excluded from the cohort. So we do actually,
Okay.
I think we do have access to those, but we didn't include that in the study.
Okay,
So,
'cause it might be
yeah.
interested to see the classification on those reports.
Yeah, definitely. That's a good point. Yeah, for a follow-up, that might be very interesting to look at.
And like, what were the reason of disagreements?
Well, I think usually what happens, so there is a consensus diagnosis from the physician. So maybe if one physician, or if they can't agree to consensus, it's essentially marked as uncertain. Right?
Okay, so if one doesn't agree, then
I'm not exactly sure
okay.
specifically about how much you need for consensus, but that's generally, from what I understand, the procedure.
Did you remove things like the age and rotation?
Yeah, so things like age information were removed. Yeah, then like the names, anything like that could be identified with them. Even things like what school they go to, this sort of thing. So it's really just like all that was left within the
It seems to me that removing the age, that might hurt your performance a little bit.
Yeah, that's true. That's true. I mean, at the end of the day, though, I think these models are so powerful, they might just be able to pick out the age and use that as a diagnostic indicator, right? But where we wanted to get at was just what things in addition to age, right? Like what are actually the patterns of behavior necessarily. So we tried to make it essentially as straightforward for the behavior parts as possible.
Thank you. Yeah, I was just curious, you don't have any recording of what happened during the panel when they discuss the case in order to take a decision, because this would be a good ground truth about the arguments which are used by the different experts.
The reports that we had, obviously, but it might be, that might be recorded somewhere that we could take advantage of. I'm not exactly sure how they do that, but that is a good point that, yeah, for actually deciding, whether one individual has autism or not, it might be interesting to look at, yeah, as you say, the consensus and how they reach that consensus. Yeah.
And in your experiment, you just have a binary decision, autism, not autism.
Yeah.
I suppose it's more complex.
Yeah, definitely. Of course. Yeah, it's much more complex than that in reality. But we really wanted to try and get at the notion of what things are most autism specific. And again, like a follow-up study might look at more of those, fine-grained details, like is this, definitely autism or maybe that sort of thing. So, but yeah, we just wanted to look at the most autism-specific indicators for sure.
Thank you.
So I don't know if you hear about Evidence Analytics. Evidence Analytics is a company here at Milo, and they are working on, they developed already a chatbot for autism children and students with the Ministry of Education. And they are working very hard to use typically NLP on, try to help students to, for dropout problem, to help students to be more integrated in schools. And there's many things that they have a very, very nice group working on autism. And they're also a member of Observatoire Québécois de l'Autisme. And there's many things they are doing here. They are here just at Mira. I don't know if it will be interesting.
Well, yeah, I wasn't actually aware of that. That's very interesting. Yeah. Yeah.
So
Yeah, it could be very interesting to work with them,
Exactly.
actually. Yeah.
yeah, I just sent them when you are presented. Just send them your presentation.
Yeah.
Hopefully
No,
they-
that's super cool. I wasn't aware of them. Yeah.
okay. So yeah.
Any other question? Let me check if there's some online.
Anything online or not yet? Okay.
No question.
All right.
All right. Well, then let's thank Jack again for the presentation.
Thanks very much. All right.